Hello, my name is Katie Hallinan, and my project is on the role of MED1 on macrophages in mammary tumors. The goal of my project was to elaborate on the role of a protein called MED1 in promoting cancer development by altering the recruitment of macrophages to a tumor tissue in a mouse model of breast cancer called MMTV-PYMT. To better understand this project, here is a little bit of background information. The Zhang lab is interested in MED1 because MED1 is overexpressed in 50% of breast cancers and contributes to overall worse patient outcomes. Functionally speaking, MED1 and its partner estrogen receptor alpha, or ER, interact via a domain on MED1 called the LXXLL motif, and they work together to promote breast cancer progression by regulating gene expression. Previous unpublished work in the lab has found that when MED1's ability to interact with ER is disrupted by a mutation in this LXXLL motif in the PYMT model, cancer progression is slower and occurs later, but this phenotype remains poorly studied. My project in the lab was to test whether changes in the recruitment of cells called macrophages may be involved. So what are macrophages and why do we care about them? First, macrophages are a type of immune cell that typically surround and kill foreign cells. There are two relevant types of macrophages when it comes to cancer progression, M1 and M2. M1 macrophages function as one would expect an immune cell to work, by assisting with killing tumor cells and preventing cancer progression. However, when macrophages come into contact with tumor cells, these tumor cells can force them to become M2 macrophages, which are cancer-promoting, and when they are found in tumors are often an indicator of poor overall prognosis. Previous work in the lab has connected MED1 and macrophage recruitment in a model of HER2-driven breast cancer. MED1 overexpression occurs in nearly all HER2 overexpressing breast cancer in patients. Further, when MED1 was overexpressed in a mouse model of HER2-driven breast cancer, tumor growth was both faster and macrophage recruitment increased compared to the HER2 model alone, which can be seen by the blue and green bars on the graph to the right. Conversely, when MED1 LXXLL motifs were mutated in the HER2 model, tumors grew slower and fewer macrophages were recruited to the tumor, which can be seen by the red bar on the graph. This indicates that MED1's role in driving growth in the model of HER2 breast cancer may be related to altered macrophage recruitment. Whether this is true in the PYMT model, however, was unclear and was the main point of investigation for this project. Here is the method that I used. On the left is a graphic of the workflow used to measure macrophage infiltration in tumors. To summarize, I took preserved tumors and cut them into sections approximately 1 cell layer thick. After verifying the quality of the tissue, these samples were incubated with antibodies which specifically labeled the macrophage biomarker called F480. Next, I used a hematoxylin counterstain that was used to stain all the nuclei in the sample to compare. In the example image, you can see the macrophages are stained brown, while all the background cells and nuclei are stained purple. The number of macrophages per number of total nuclei were then counted from the images taken to obtain a percentage of macrophages for each tumor. The results from the counts taken for each tumor type show some of the trend we were expecting to see. There were generally fewer macrophages counted in the MED1 knockdown tumors compared to the heterozygote with functioning MED1 expression. While we need additional data points to perform a statistical analysis, these preliminary results suggest that MED1 may be playing a similar function in this PYMT model as was previously discovered for the HER2 model. In conclusion, while we still need additional samples to finalize our experiment, our preliminary data suggests that MED1's interaction with ER may be necessary for macrophage recruitment. After gathering additional data, it will also be essential to use additional biomarkers to analyze whether these macrophages are M1 or M2 in character. I would like to thank the Zhang lab, especially Gregory Bick and Jennifer Tranwin for all their help this semester. And here are the references used for this project. Thank you.